Hello students. So we shall continue with the next unit, that is biotechnology, and in that we shall consider chapter 11, that is principles and processes of biotechnology. So here biotechnology, if you divide this term into two parts, it is bio and technology. So biotechnology is the fusion of biology and technology, right? So biology and technology, when they are combined, you get biotechnology. So what is this biotechnology? So biotechnology, it is the, you can say, field that mainly uses live organisms, right? So it is the technology that uses live organisms to produce products which are important to man. So that is the meaning of biotechnology. So biotechnology is the technology which uses live organisms to produce products of human importance. So which products are produced by biotechnology? So what are some of the products which are produced by this technology? So one is hormones. Different hormones are produced by biotechnology. Then enzymes. Various enzymes are produced by biotechnology. Then protein. Protein is produced. So all these can be called as the products of biotechnology, which can be produced by using live organisms. So the commonly used organism here, it is the bacteria, mainly E. coli, Escherichia coli, or in short E. coli. So E. coli is a bacterium commonly used in biotechnology, right? So that is the, you can say, organism which is commonly used in biotechnology, right? So live organisms. The products, these are the different products. So one example we shall take, one example of hormone, right, that is produced by biotechnology. Again you will be studying how this hormone is produced. So that hormone is insulin. So insulin is one example of hormone that is produced by biotechnology, right? So you will be studying synthesis of insulin by biotechnology in the coming part. So insulin is a hormone, right? It is a peptide hormone. Peptide hormone means the hormone that is made up of protein. It is called peptide hormone. So insulin performs a important function in our body. So what is the role of insulin in our body? So insulin is involved in controlling glucose levels in our body. So insulin controls glucose levels, right, in our body. So that is the main function performed by insulin, right? So imagine glucose is more in our body, or glucose levels are very high in the blood. This excess glucose is converted into glycogen, right? So converting excess glucose into glycogen is done by insulin. So in this way insulin helps to control glucose levels by converting excess glucose into glycogen, and that glycogen is stored in the liver cells, right? So that is the brief introduction about the biotechnology. The next we will consider definition. So definition of biotechnology, which was given by EFB. So here EFB indicates European Federation of Biotechnology, right? So EFB indicates European Federation of Biotechnology. So this federation, European Federation of Biotechnology, right, gave a definition for biotechnology. So this is the definition. So what this definition says, the integration of natural sciences, right, the integration of natural sciences, organisms, cells, parts thereof, and molecular analogs, right, and molecular analogs for products and services. So this is the definition of biotechnology given by EFP. So you may expect a 1 or 2 mark question. Write the definition of biotechnology as given by EFB. So this definition you have to write. So integration of natural science, organisms, cells, parts thereof, and molecular analogs, right, for products and services. So all these are used, right? Integration means to unite or to combine or to use, right? So use of natural science, use of organisms, use of cells, their parts, use of molecular analogs, all these are integrated or used mainly for producing products, right? So these are the products, hormones, enzymes, proteins, etc. Fine. Molecular analogs. So what are these molecular analogs? So a molecule, right? A molecule which is similar to another in structure, right? So one molecule is similar to another molecule in its structure. They are called molecular analogs, right? So this is the definition of biotechnology. Then the process involved or included under biotechnology. So biotechnology includes, right? Biotechnology includes different processes or different techniques. So which are those? So some are listed here. One is IVF. So IVF is one process included under biotechnology. IVF, the full form, in vitro fertilization. So this is the full form of IVF, that is in vitro fertilization, right? So in vitro means in a apparatus, in the laboratory, outside the body, right? So any, you can say, process that occurs in the lab, normally done outside the body, it is called in vitro. Fertilization, that is fusion of male and female gametes. So fusion of male and female gametes is done in the lab, in the apparatus, or in a test tube or in a flask. It is called in vitro fertilization, which is also called test tube baby. So in vitro fertilization is also called test tube baby. So this is one technique or process that is included under biotechnology. Then the next one is gene or DNA synthesis. So synthesis of genes or synthesis of DNA also comes under biotechnology, right? So how these genes or DNA, see, a gene and DNA both are same, how they are synthesized. So synthesis of a particular gene or DNA, it can be done by linking nucleotides, right? So here the nucleotides are created first, and these specific nucleotides, they are linked, right? They are linked by bonds, right? They are linked by bonds, and DNA or gene is produced, right? So this process is called DNA synthesis, which is carried out in the lab, right? So in the biotechnology lab, linking of nucleotides by bonds produces gene or DNA, and that can be used for different techniques or processes. So gene or DNA synthesis is one process included under biotechnology. Then developing DNA vaccines. So DNA vaccines are developed, right, in the biotechnology labs. So synthesis of DNA vaccines is another process that comes under biotechnology. So DNA vaccines, these vaccines, they contain DNA, right? They contain DNA of a particular microbe, that is DNA of bacterium or DNA of virus, right? So such vaccines which have DNA of microbes like bacteria and virus, right? They are called DNA vaccines, and these vaccines, they increase the immunity, right? The immune power or immunity is increased by these DNA vaccines, and these vaccines help in preventing diseases. Then gene therapy, right? Correcting a defective gene, right? It is called gene therapy. So therapy means treatment. Therapy means treatment. So treatment of genes. Which genes? Defective genes or faulty genes. The genes having a defect or a fault. Such genes are treated by a technique called gene therapy. So all these processes, they come under biotechnology. Then next we shall consider the core techniques that enabled the birth of modern biotechnology. So core techniques means the main techniques, right? The main techniques that are responsible for the birth of modern biotechnology. So the two core techniques, right? The two core techniques are, one is the genetic engineering and second is chemical engineering. So genetic engineering, GE, right? Genetic engineering and CE, that is chemical engineering. So these are the two core techniques that enabled or that made possible the birth of modern biotechnology. So what is this genetic engineering? So genetic engineering, it is the technique, right? It is the technique of alteration of genes or DNA, right? So alteration of genes or modification of genes is done by a technique called genetic engineering, right? Then what is done with that modified gene? See, a gene is modified or DNA is modified. That modified DNA, now it is introduced in an organism. So what happens? The phenotype of that organism changes. So there is change in the phenotype of organism, right? So all these techniques, they come under genetic engineering. Then chemical engineering. So chemical engineering, it deals with the, you can say, creation of sterile environment, right? So a sterile environment is created which favors the growth of particular microbes, right? So this is very important in biotechnology. Sterile conditions are created, and these sterile conditions, what they do, they favor the growth of only desired microbes, right? Only selected microbes, they can grow in such sterile conditions, and development of sterile conditions is done with the help of chemical engineering. Now the microbes which are grown in sterile conditions, those microbes, they produce protein, and that protein is used to produce products, right? So these two core techniques enabled the birth of modern biotechnology. The next we shall consider the oocyte. So here oocyte, it also means the origin of replication. Oricite means origin of replication. So this is a site, right? Ori is a site present on DNA which initiates replication. Which initiates replication. What is the meaning of replication? Replication means duplication or multiplication or reproduction, right? So this site, it initiates or it starts the process of replication of DNA or replication of desired gene, right? What is the meaning of desired gene? See, a desired gene is also called gene of interest. Desired gene is also called the gene of interest, right? So, desired gene or gene of interest is replicated, right? It is replicated to produce many copies. So, this process of replication of desired gene is initiated or controlled by this site located on DNA, which is called Ori site. So Ori site means origin of replication, right? Which initiates replication process. Then gene cloning. So gene cloning is a process of producing multiple copies of desired gene. The process of producing multiple copies of DNA or gene, it is called gene cloning. So, if there is a single gene, many copies of that gene can be produced, right? So, many copies of gene can be produced, and that process to produce multiple copies of a gene, it is called gene cloning, right? So, gene cloning here is done by using certain, you can say, microorganisms like bacteria. How it is done, we shall discuss in the coming part. So that is about the today's video. So in today's video, we have discussed the meaning of biotechnology. Then we discussed the definition of biotechnology, which was given by the European Federation of Biotechnology, right? EFB. Then we discussed different processes that are included under biotechnology. Then we considered the core techniques that enabled the work of biotechnology, right? Such as genetic engineering and chemical engineering. And then next we discussed Ori site or origin of replication. And finally, the last part was gene cloning. So, we shall be using these terms in the coming concepts, right? So, try to be familiar with all these terms, right? The terms such as genetic engineering, the terms such as gene therapy, the terms like Ori site, then gene cloning, etc. Right? So we shall conclude here and we shall continue in the next video. Thank you.